Q1. What is real-time PCR? Real-time PCR is a molecular diagnostic technique that enables the detection and quantification of DNA or RNA in real time during the PCR reaction. Q2. How does real-time PCR work? Real-time PCR involves the use of fluorescent probes that bind to the amplified DNA or RNA during the PCR reaction. The fluorescent signal is measured by a detector in real time, allowing the quantification of the target sequence. Q3. What are the advantages of real-time PCR over traditional PCR? Real-time PCR is faster, more sensitive, and more specific than traditional PCR. It also allows for the quantification of the target sequence, which is not possible with traditional PCR. Q4. What is the role of real-time PCR in diagnostic virology? Real-time PCR is widely used in diagnostic virology to detect and quantify viral nucleic acids in clinical samples. It is a sensitive and specific method for the diagnosis of viral infections. Q5. What are the limitations of real-time PCR in diagnostic virology? Real-time PCR can be affected by inhibitors present in clinical samples, leading to false negative results. It also requires specialized equipment and expertise, which may not be available in all diagnostic laboratories. Q6. What are the types of fluorescent probes used in real-time PCR? The two main types of fluorescent probes used in real-time PCR are TaqMan probes and SYBR green probes. Q7. What is the difference between TaqMan probes and SYBR green probes? TaqMan probes are specific to the target sequence and emit fluorescence only when they hybridize to the target, whereas SYBR green probes bind to any double-stranded DNA and emit fluorescence when intercalated between the DNA strands. Q8. How is real-time PCR used in the diagnosis of viral infections? Real-time PCR is used to detect and quantify viral nucleic acids in clinical samples such as blood, urine, and respiratory secretions. The results are interpreted based on the threshold cycle, CORT, value, which indicates the amount of viral nucleic acid in the sample. Q9. What are the advantages of using real-time PCR for the diagnosis of viral infections? Real-time PCR is highly sensitive and specific, allowing for the detection of low levels of viral nucleic acid in clinical samples. It is also faster than traditional viral culture methods and can provide results within a few hours. Q10. How is real-time PCR used to monitor viral load in patients undergoing antiviral therapy? Real-time PCR can be used to monitor changes in viral load over time in patients undergoing antiviral therapy. A decrease in viral load indicates a response to therapy, whereas an increase in viral load may indicate treatment failure. Q11. What is the significance of the CORT value in real-time PCR? The CORT value is the cycle at which the fluorescent signal generated by the probe exceeds a predetermined threshold. The CORT value is inversely proportional to the amount of viral nucleic acid in the sample, with lower CORT values indicating higher viral loads. Q12. What is the limit of detection of real-time PCR for viral nucleic acid? The limit of detection of real-time PCR for viral nucleic acid is typically in the range of 10 to 100 copies per reaction, depending on the assay design and sample type. Q13. How can real-time PCR be used to differentiate between viral strains or subtypes? Real-time PCR can be used to design specific probes or primers that target conserved regions of the viral genome, allowing for the detection and differentiation of viral strains or subtypes. Q14. How can real-time PCR be used to monitor the emergence of drug-resistant viral strains? Real-time PCR can be used to detect mutations in the viral genome associated with drug resistance. Monitoring viral load and drug-resistant mutations over time can help guide treatment decisions. Q15. What are the challenges associated with using real-time PCR for the diagnosis of viral infections in resource-limited settings? The high cost of real-time PCR equipment and reagents, as well as the need for specialized expertise, can be a challenge in resource-limited settings. In addition, the lack of access to reliable electricity and laboratory infrastructure can also be a barrier. Q15. What are the challenges associated with using real-time PCR for the diagnosis of viral infections in resource-limited settings? The high cost of real-time PCR equipment and reagents, as well as the need for specialized expertise, can be a challenge in resource-limited settings. In addition, the lack of access to reliable electricity and laboratory infrastructure can also be a barrier. Q16. What is real-time PCR, and how does it work? Real-time PCR, also known as quantitative PCR or qPCR, is a molecular biology technique that allows for the detection and quantification of specific DNA sequences in real time during the amplification process. It works by monitoring the fluorescence emitted by a dye that binds to the double-stranded DNA, dsDNA, product generated during PCR. As the amount of dsDNA increases with each PCR cycle, the fluorescence intensity also increases, and this can be measured by a detector to calculate the amount of target DNA present in the original sample. Q17. What are the components of a real-time PCR reaction, and what are their functions? Q18. How does real-time PCR detect and quantify the amount of target DNA in a sample? Real-time PCR detects and quantifies the amount of target DNA by measuring the fluorescence intensity of a dye that binds to the dsDNA product during each PCR cycle. At the beginning of the reaction, the template DNA is denatured, i.e., separated into single strands, at a high temperature, typically 95 degrees Celsius. Then, the primers anneal to the target sequence at a lower temperature, typically 55 to 65 degrees Celsius. The DNA polymerase enzyme extends the primers to synthesize new DNA strands that are complementary to the template DNA. As the PCR reaction proceeds, the amount of dsDNA product increases exponentially, and this is monitored by measuring the fluorescence intensity of the dye after each temperature cycle. The amount of target DNA in the original sample is calculated based on the cycle threshold, CORT, value, which is the cycle number at which the fluorescence signal exceeds a set threshold level. Q19. What are the advantages and limitations of real-time PCR compared to other PCR methods? The advantages of real-time PCR include its high sensitivity, specificity, and accuracy, as well as its rapid turnaround time and ability to detect and quantify multiple targets simultaneously, in multiplex PCR. It is also less prone to contamination than other PCR methods that require post-amplification analysis. However, real-time PCR requires more specialized equipment and reagents than conventional PCR, and it can be affected by inhibitors in the sample that interfere with the PCR reaction or fluorescence signal. In addition, it is typically more expensive than conventional PCR, which may limit its widespread use in resource-limited settings. Q20. What are some common applications of real-time PCR in research and diagnostics? Real-time PCR has many applications in research and diagnostics, including gene expression analysis, pathogen detection, quantification of viral load, genotyping, forensic analysis, and cancer diagnosis. In research, real-time PCR is used to study gene expression patterns and identify genetic mutations associated with diseases. In diagnostics, it is used to detect and quantify pathogens, such as bacteria and viruses, in clinical samples, monitor the progression of infections, and identify genetic markers of disease susceptibility.